Our next speaker is Dr. Lili Ji. He is professor and director of the School of Kinesiology at the University of Minnesota, Twin Cities. Dr. Ji received his MS and PhD degrees at the University of Wisconsin-Madison, specializing in exercise physiology. His postdoctoral training was in biochemistry at the Institute of Enzyme Research, also at the University of Wisconsin-Madison. Dr. Ji's research focuses on cellular and molecular exercise physiology, especially in the role of free radicals and antioxidants in muscle dysfunction, adaptation, pathogenesis, signaling, and aging.
Well, thank you. Life is very tough. We're trying every way to reverse the natural cause imposed by aging. And some are effective, successful, and some are not. We have heard the great story by Dr. Barnes and Dr. Sears on the exercise effect on brain, vascular system. They have alluded to the detrimental effect of reactive oxygen species. I'm going to talk in the other phase of the reactive oxygen species, which is called redox signaling. We all believe exercise is a medicine, and what we want to know is why and how. So we're going to look microscopically into the cell to find out some of the beneficial effect of reactive oxygen species in causing some of the adaptations that we have learned. What is redox signaling? This is really a kind of overwhelming summary of the properties of redox signaling, and I'm hoping in the end of the talk we'll have a little understanding of all the things. Generally speaking, redox signaling is the signal transduction that relies on some of the redox mechanisms in the cell. It's being done by some of the signals produced either outside of the cell or inside of the cell. These pathways are usually, but not always, reversible, and due to a number of chemical mechanisms such as phosphorylation, acetylation, and sulfur hydromodification, and others. And one of the common features is, in the end, we need some chemical or physical mechanisms to modify the transcription factors finally required to bind with the DNA, confer some of the effect. This is really just an outline. And what I'm going to talk is first we're going to give some general introduction of what redox signalings are, and then we'll move on to talk some of the specifics about aging and pathogenesis. I'm going to focus on is muscle. So it's going to be muscle atrophy caused by inactivity. And in the end, I'm hoping to provide some insight of what ways we can do to prevent this from happening. We learned from general physiology that there are several ways that can confer cell-to-cell communication. The most recognized way is the endocrine system, and that's depend on blood-borne substance. The second way is the neural system. And what I'm going to talk about is the called autocrine or paracrine system that is relying on the locally produced products. And in this case, it's the reactive oxygen species that serves as the signal molecule. So in order to do signaling, we need to have a signal. So where is the signal coming from? And we have talked, Dr. Sears provided a very general introduction of the reactive oxygen species. We have several ways in the cell that would produce reactive oxygen species during exercise. One of the most common way we all know is from the mitochondria. It's just the oxygen consumption. And it's particularly true during aerobic exercise. The second is the xanthine oxidase that is activated when the exercise is hypoxic, particularly during the reoxygenation or reperfusion phase. That enzyme is activated to produce superoxide. The third is a membrane mechanism caused by cyclic oxygenase or lipooxygenase. And they're usually activated by pro-inflammatory cytokines. Certain cytokines can activate this pathway. And the fourth is nitric oxide, NADPH oxidase. In this particular, this enzyme is present in many different places, but what I'm talking about here is that in the neutrophil, when muscle is being injured, these cells will infiltrate into the cell and trigger a respiratory burst that causes the release of reactive oxygen species. This is more prevalent after exercise and after damaging exercise rather than during exercise. So overall, These provides a general view of what the sources are during exercise. After we have the signal, we need to know what is the target. And in order to understand that, we need to know a little bit about the DNA structure. DNA is a double helix made of nucleotides. Because of the specific nucleotide, when they wind up, they form a larger gap between the two. It's called a major groove. And on the opposite side is the minor groove. And because of the different chemical bonds, and the major groove provides more rich opportunities for proteins to bind. Major groove usually can form the area of a promoter. Promoter is the area prior to the activation of transcription sites that is rich in the area called DNA binding sites or nucleotide regulatory sequence. And then we also need to have the chemicals that bind with the DNA, and those are called transcription factors or DNA binding proteins. And they have the structural motifs that can recognize the DNA binding sites. Oftentimes, these transcription factors require chemical modification or dimerization or dissociation between the transcription factors to make them capable of binding. So these are some of the possibilities of how proteins can bind with the DNA. If we look closely, we can see that this is the major groove area, that we have some of the hydrogen bonds or nitrogen bonds that are recognized by a part of the protein that causes these binds. So this is just a few of the larger possibilities. Usually it contains about 10 to 20 such sequences in order to be recognized by the transcription factors. So we mentioned that various ways during exercise could cause reactive oxygen species generation. And in the end, these reactive oxygen species will converge to hydrogen peroxide, as SOD will turn superoxide into hydrogen peroxide, and many of the membrane bond enzymes will activate hydrogen peroxide activation. And despite the fact that there are antioxidant enzymes available, if the exercise is intense enough, that could break the balance and result in a surplus of hydrogen peroxide. And hydrogen peroxide turned out to be the most important signal in the redox signaling for a number of reasons. Because it's small enough, it could diffuse, it's relatively stable, it is oxidative, but yet not highly damaging, making it an ideal signaling molecule to deliver the signal to the nucleus to cause some of the redox signaling. And what I'm going to focus on are the three major pathways: NF-kappa B, MAP kinase, and PGC1 alpha. DNA, as I mentioned, contain promoter regions that are rich in DNA binding sites, and they could be recognized by the various transcription factors. Usually, they are located in the cytosol, but upon the activation of signaling, they can migrate into the nucleus and find a way to bind. Some of the intron regions, that is the DNA sequence not coded for a particular protein, also contain these binding sites, and they could also be recognized by the transcription factors. It is the combined effect. So each of these transcription factors possibly are required, but it's not sufficient to induce the activation transcription. So it is the coordinated effort of various pathways that would merge and produce an effect that is synchronized to activate the gene expression. First, let's focus on NF-kappa B. Dr. Sears mentioned NF-kappa B is an active and pro-inflammatory pathway. It is true. NF-kappa B is a trimer that contains a I-kappa B site, two subunits called P65 and P50, and usually they are sequestered together in the cytosol. But when the enzyme that is phosphorylating the I-kappa B subunit, then trimer is dissociated, and P65 and P50 are released into the nucleus and can bind with the DNA. The enzyme that activates this process is called IKK, I-kappa B kinase, and also the upstream NF-kappa B induced kinase. And these two kinases are redox sensitive. They could be activated by MAP kinase, which was in turn activated by PKC, a very important commanding enzyme in a MAP kinase pathway that I'm going to talk about. So what is the evidence exercise can activate NF-kappa B? If a rat is being exercised for an hour, it produces sufficient amount of hydrogen peroxide to activate these two enzymes. And there are a couple of ways the enzyme could be activated. One is a direct activation by PKC, by hydrogen peroxide. Hydrogen peroxide also oxidizes an enzyme that is called phosphatase. And normally, phosphatase keeps the enzyme in a balanced status. So any activation could be reversed by the phosphatase. But when hydrogen peroxide oxidizes and inhibits the phosphatase by sulfur hydroxidation, then this enzyme is inhibited so that any small signal can cause a larger and sustained activation of these two enzymes, causing a sustained NF-kappa B activation. When a drug called PDTC is used to remove the hydrogen peroxide and inhibit NF-kappa B, this effect is attenuated. A toxin called the lipopolyoxide, LPS, can also mimic the exercise effect by directly activating MAP kinase. To show that it's only the hydrogen peroxide, but not any reactive oxygen species can activate the NF-kappa B, we could use another oxidant called T-butyl hydroperoxide. It doesn't produce an effect to mimic the exercise effect. It is not any of the reactive oxygen species, but only hydrogen peroxide that can activate NF-kappa B. The I-kappa B is phosphorylated in this case, and it migrates to the nucleus, and we can see that increased P65 presence in the nucleus. And the time course is about 1 to 2 hours after the exercise. That is the time required to have all this occur. So NF-kappa B, whether it's effective in positive or negative, it depends on the situation we're going to talk about. But exercise will activate NF-kappa B. When it's overactivated, it produces inflammatory effect. But many of the antioxidants also require NF-kappa B activation in order to induce their gene expression. The second pathway is MAP kinase, and it's rather complicated. This is only a part of the MAP kinase family. As we said, the PKC is a commanding enzyme that could be activated by a redox mechanism, and it controls these three pathways. One is called P38, one is called JNK, one is called ERK. And their function is to phosphorylate various transcription factors that we talked before to make them capable of moving into the nucleus. And in many cases, the targeted gene requires binding of the nuclear factors activated by the MAP kinase pathway. And we also mentioned before that MAP kinase can also activate NF-kappa B pathway. This study done in Harvard University in the last 10 years indicates that MAP kinase could be activated in the skeletal muscle. When muscle exercise, the P38 pathway is activated. If we use a ERK, is activated. If we use an ERK inhibitor, that effect is gone. But if we use a P38 inhibitor, that doesn't take effect away. When these two are combined, of course, you show the ERK effect by combined inhibitor. P38 is also activated. ATF2 is a transcription factor controlled by the P38 pathway. And as we can see that P38 inhibitor takes away some of the effect, but not ERK effect. This study shows that MAP kinase is indeed activated in skeletal muscle. We also used a rat model to indicate that if rats are sprinting, the enzyme xanthine oxidase is activated. If we use a inhibitor called allopurinol to decrease xanthine oxidase, the activation of NF-kappa B by exercise is removed. When we use the xanthine oxidase inhibitor, P38 activation is removed. MAP kinase is a redox-sensitive pathway that is activated during exercise and controlled by a redox mechanism. And this shows that the two ERK enzymes, ERK1 and ERK2, also activated by exercise and could be prevented by allopurinol administration. Now we're moving to the next pathway called PGC1 alpha. This is a nuclear cofactor, so it doesn't directly bind with the DNA, but is required in many signaling pathways, including ERR, estrogen receptor-induced effect, PPAR-induced effect, and this nuclear cofactors could be produced by exercise due to a number of different mechanisms, including calcium, reactive oxygen species, nitric oxide, and so on and so forth. And this cofactor is being shown to play a major role in causing the training adaptation to aerobic exercise, such as fiber transformation. PGC1 alpha is also an important requirement for antioxidant expression. So in this study, and we can see all the major antioxidants in the cell could be activated by hydrogen peroxide treatment. But when the PGC1 is knocked out, these effects are gone. So PGC1 is important in many of the cellular response to exercise. Recently, it's also been shown that a subtype of PGC1 is called PGC1 alpha 4. A shorter version of PGC1 is important in causing muscle hypertrophy caused by resistant exercise. So when PGC1 alpha is intact, it induces oxidative adaptation, muscle, increased mitochondria, and so on and so forth. But when it's a shorter version, it induces hypertrophy. So you can see the two major exercise effects could be benefited from PGC1 induction. PGC1 alpha needs to be both expressed and also phosphorylated before it can bind with the other transcription factors. And as I mentioned in the previous one, contraction causes calcium release, which activates CAMK and calcineurin that are directly affecting PGC1 expression. Catecholamine, to some extent, can also activate the pKa and also play a role in phosphorylating CRAB, which binds with the promoter region of PGC1. But also importantly, reactive oxygen species, either through xanthine oxidase or through mitochondria, can activate PKC and through a P38 mechanism influence the expression and the phosphorylation of PGC1. When PGC1 binds with its cofactors such as NRF, nuclear respiratory factor 1, it can induce the transcription of the nuclear encoded mitochondrial components that moves into the mitochondria, such as electron transport chain, most of the electron transport chain subunits. But also, it can induce a very important enzyme called mitochondrial transcription factor A, or TFAM. This transcription factor promotes the DNA replication in the mitochondria to make more mitochondria. So these two are both important in causing the mitochondria proliferation and adaptation in response to aerobic exercise and other stimuli such as cold exposure, stress, and so on and so forth. PGC1 alpha pathway could be activated by an acute bout of exercise as well as chronic training. So when rats are sprinting on the treadmill for a protocol lasting about 40-50 minutes, it has a dramatic effect on the gene expression of PGC1 alpha. If during the sprinting exercise we use allopurinol again, we can inhibit about half of the exercise effect. And this shows that allopurinol knocks down the xanthine oxidase and decreases reactive oxygen species production. And this effect has been shown to be caused by, in part, by CRAB inhibition. So when exercise-induced CRAB phosphorylation is activated, it could be prevented by allopurinol inhibition. And also, CRAB, as I mentioned just before, is controlled by P38 pathway. So exercise-induced P38 activation is partially inhibited by the removal of some of the reactive oxygen species. If we believe those three pathways are activated by exercise, what is the physiological consequence? We say one of the major effects of redox signaling is to induce antioxidant defense. As Dr. Sears provided some evidence in the vascular system. We're talking about muscle here. Antioxidants are plays an important role in cellular life, not only protecting against potential oxidative damage, but also control the steady state level of hydrogen peroxide, which mediates many of the proper signaling pathway. In the early last century, this group from Stanford University in Dr. Rondo's lab showed that hydrogen peroxide can activate NF-kappa B pathway in both a dose response and time-related course, and hydrogen peroxide activation induces enzyme, as shown by the reporter gene. And here, glutathione peroxidase, another important antioxidant enzyme, which removes hydrogen peroxide, is also controlled by NF-kappa B. When the I-kappa B is mutated, this hydrogen peroxide effect is greatly lost. In a physiologic context, back in the 90s, a number of groups of exercise physiologists showed that exercise can induce superoxide dismutase in both a time-related and intensity-related manner. And this activity increase could be explained largely by the gene expression of manganese superoxide dismutase, as shown in the various type of skeletal muscle. And of course, copper zinc oxide, SOD, also respond to exercise, but in aerobic exercise, it's mainly the mitochondria, MNSOD, that plays a major role. In a later study, it's being shown that an acute bowel exercise can turn on the gene of manganese SOD. So even one acute bowel exercise can cause the messenger RNA of NSOD increase by this much. But if we use allopurinol, again, to remove the hydrogen peroxide, then this exercise effect is lost. And the same thing happens to INOS, another targeted gene of exercise. Chronic training has also been shown to induce PGC1 alpha expression. So this is Dr. Hollis's lab in Wash U shows that rat swimming training could induce PGC1 alpha gene expression and also produce some of the effects in fiber transformation by another lab. Besides antioxidant enzymes, there are other antioxidants. One such example is uncoupling protein. Uncoupling protein provides a vehicle for some of the accumulated hydrogen ions in the intramitochondrial membrane to go back to the matrix so that reduce the redox pressure, if you wish, on the electron transport chain, which produces reactive oxygen species. So when exercise is imposed to the muscle, uncoupling protein can serve as a gate that opens the hydrogen ion channel. And also, the gene expression of uncoupling protein is also increased. And this is being shown through a similar mechanism as the manganese SOD, through the PGC1 as well as the MAP kinase pathway to increase the uncoupling protein. So in this particular study, exercise at various duration has been shown to increase the messenger RNA level of uncoupling 3, which is in muscle specific, and also the protein content of uncoupling protein 3 in the rat skeletal muscle. It is important to recognize that these signaling molecules play a coordinated way. It's called crosstalk, because each of the pathways may have this or that effect. As mentioned, NF-kappa B can produce both a positive or negative effect. Positive effect is some of the antioxidant systems, but it also produces inflammatory cytokines. So it needs to be kept in check. PGC1 is important in producing antioxidant enzymes that modulate the hydrogen peroxide production so that NF-kappa B activation could be kept in check. But PGC1 alpha also directly physically inhibit NF-kappa B in the nucleus, so that when you have aerobic exercise and a high expression of PGC1 alpha could produce the beneficial effect of the things that I talked about while inhibit NF-kappa B. Whereas during inflammatory status, such as negative grade walking, that is downhill walking, and muscle stretch exercise could overactivate NF-kappa B, which inhibit PGC1 alpha, so that some of the aerobic exercise produced effect is gone. So we know that during downhill running or lengthening contraction, you don't see a mitochondrial adaptation, but rather you see inflammation. So this crosstalk is very important in actually decide which effect, physiologic effect, is being put out. And also we show that the MAP kinase pathway activates NF-kappa B, whereas hydrogen peroxide activates PKC. So the interaction of the various pathways is the most important rather than the individual intensity of each of the redox pathways. So in order to know this crosstalk, we designed a study where the drug PDTC is used to treat the rats which are also endurance exercised. So you can see that endurance exercise produced effect of NF-kappa B activation is inhibited by the drug. Also, the crab binding is inhibited by PDTC, which is NF-kappa B inhibitor, also inhibited crab binding, which is a PGC1 alpha produced effect. And PGC1 alpha itself is increased by endurance training, but when PDTC, NF-kappa B inhibitor is imposed, this training effect is greatly lost. And the cytochrome C oxidase is increased by endurance training, but when NF-kappa B inhibitor is used, that training effect is also lost. NF-kappa B could inhibit PGC1 alpha induction caused by endurance training. So as the first summary, we say that moderate level of ROS surplus may activate redox signaling, and this is mainly caused by activation of NF-kappa B, MAPK, and PGC1 alpha. And the net result is the stimulation of antioxidant gene expression, and crosstalk of multiple signaling pathways are required to confer the transcriptional activation of target gene, and the physiological outcome of exercise may be determined by the relative intensity of the various signaling pathways, as we will show later in the muscle immobilization situation. So in the second part, I'm going to talk about the impact of redox signaling on aging and muscle atrophy. So in the second part, I'm going to talk about the impact of redox signaling on aging and muscle atrophy. We know that aging could cause a steady decline of muscle mass and a force production called sarcopenia, and decreased mitochondria population and oxidative phosphorylation capacity is an important mechanism for the muscle loss. And we hypothesize that a downregulation PGC1 alpha signaling may be an important mechanism for this pathology. So this shows that age-dependent decrease in the mouse muscle in state 3 and state 4 respiration. And this could be prevented in part by aerobic training. When animals get older, we can see that both the messenger RNA levels and the protein levels of PGC1 alpha is decreased. When rats are subject to aerobic training, much of the aging effect could be reversed. This may be looking a little dramatic, but it shows that so-called aging effect may be in part due to the sedentary lifestyle, as the muscle is really immobilized in the cage when the rats living in a sedentary life. Exercise could be a major mechanism to reverse this so-called aging effect. This is some of the effects, such as TFAM, the mitochondria, DNA to nuclear DNA ratio that's been shown decreased by aging, but reversed by exercise training, and both at the mRNA level and at the protein level. Again, much of this effect is caused by the signaling pathways controlled by P38 and PK that is shown to respond to exercise training even at old age. And this shows the binding of CRAB to DNA by a gel mobility shift assay, shows that DNA binding of CRAB is dramatically decreased. And because CRAB is the upstream controller of PGC1 activation, so it shows that this weakened binding during aging may be important to explain some of the loss of PGC1 alpha and oxidative capacity, whereas training can reverse that trend. SERT is a deacetylase that deacetitate the PGC1 molecule. So when PGC1 molecule is acylated, it loses its function. So SERT can remove the acetate from the PGC1 molecule to partly reverse its function. So you can see that exercise training also increases the SERT activity. Mitochondria constantly goes through division and fusion. So it's called mitochondrial dynamics. Mitochondria could fuse together or could split out. So this is controlled mainly by the fusion protein and the fission protein. And aging has been shown to decrease mitochondrial fusion protein expression, whereas increase mitochondrial fission expression. So mitochondria gets more fragmented during aging. And exercise training has been shown to increase the fission protein expression, and the fission protein fusion protein expression has a positive correlation with the PGC1 alpha. And it turned out to be that the fusion protein 2 is also a target gene for PGC1 alpha. So age-dependent loss of the fusion protein is reversed by exercise training, and exercise training also increases PGC1 alpha expression in the red skeletal muscle. Recently, it's been shown that overexpression of PGC1 alpha through transgenic method could reverse many of the dramatic effect of aging in the skeletal muscle. When mouse were overexpressed by PGC1 alpha, the loss of mitochondrial functions such as COX and cytochrome C oxidase, cytochrome C protein levels are reversed by overexpression of PGC1 alpha. And also overexpression of PGC1 increases SOD gene expression. On the other hand, age-related increase of pro-inflammatory cytokine expression is prevented or decreased by overexpression of PGC1. The impact of signal transduction on aging could be explained by the mitochondrial deterioration by aging, and the downregulation of PGC1 alpha has been shown to play a major role. Both endurance exercise and transgenic overexpression can ameliorate age-related decline of mitochondrial function. Muscle immobilization could also cause muscle atrophy, and this is very relevant in exercise situation because muscle injury, bed resting, and traumatic effect, or even microgravity can cause muscle atrophy. Muscle protein loss could be caused by both decreased synthesis and increased degradation. In the case of muscle disuse atrophy, enhanced proteolysis is the major reason. And there are two ways to control signaling of muscle proteolysis. One is the NF-kappa B controlled ubiquitin proteolysis. One is the FOXO controlled lysosome autophagy. And these two are playing different roles, but probably all important in causing muscle atrophy. And this is the model that shows healthy muscle volume is maintained by the AKT mTOR pathway that inhibits the FOXO, whereas the muscle inactivity could cause NF-kappa B activation and cause reactive oxygen species overexpression. They would activate two specific muscle proteolytic pathways that is related to ubiquitin, eventually activating two enzymes. One is called HG1, one is called MRF1, and they target specifically to muscle proteins such as Z-lines and troponin, major troponin components, and actin. And the PGC1 alpha has been shown to inhibit both NF-kappa B and also FOXO, so that PGC1 alpha may play a central role in muscle atrophy. That's our hypothesis. So by using a mouse hind limb banding model, we could produce about 15% of the hind limb muscle atrophy in both tibialis anterior and gastrocnemius. When you sort out the messenger RNA for the various oxidative components, we can see that 1 week, 2 weeks, 3 weeks of immobilization could cause a major decline of many of the mitochondrial components, whereas 1 week remobilization and 2 week remobilization, really it's 2 weeks that could cause a more complete recovery, whereas 1 week doesn't produce enough effect. So we used a model of having muscle immobilized for 2 weeks and then remobilized for 1 week to observe what happens to these signaling pathways and how they control the outcomes. So PGC1 alpha protein level lost by 50% after 2 weeks of immobilization. And after 1 week of remobilization, there's no recovery. And this is caused by a dramatic loss of both NRF1 and NRF2. And again, remobilization is not sufficient to recover. When you look at the mitochondria, various mitochondrial structural and functional components, you can see that TFAM, COX, and cytochrome C, and they except the cytochrome C oxidase subunit 4, immobilization caused a dramatic decrease of the mitochondrial components. When you look at the mitochondrial DNA to nuclear DNA ratio, it's dramatically decreased. And the mitochondrial COX level, activity level is dramatically decreased. And also, the ATP production rate in the mitochondria is decreased. And none of these could be very effectively reversed by 1 week of remobilization.
Okay, great.
We further look at the effect of immobilization on inflammatory cytokine expression, and we can see that 2 weeks of immobilization really increases all the major pro-inflammatory cytokines, such as TNF-alpha, interleukin-1, interleukin-6, only after 2 weeks that they can recover, whereas the 1 week remobilization is actually cause an increase rather than decrease of pro-inflammatory cytokine expression. And this is partially explained by an interesting increase of hydrogen peroxide during the remobilization. This is the oxidative damage, the lipid. Isoprostane is a marker of lipid peroxidation. Is increased with the increase of reactive oxygen species. And TNF-alpha and interleukin-6 are maintained at high level both during immobilization and remobilization period. And this expectedly caused a increase of NF-kappa B activation. As you can see that it's a remobilization again that increases the NF-kappa B and also the nuclear accumulation of the subunit P50. Well, we mentioned before that FOXO is a negative regulator of muscle protein. When FOXO is phosphorylated, it leaves the nucleus. It's harmless. When FOXO is dephosphorylated, it binds with the DNA and activates the estrogen-1 and the MRF1. So you can see that the ratio of phosphorylated FOX to non-phosphorylated ratio is decreased during prolonged immobilization. The initial binds of the ratio could be explained by an overexpression of mTOR, which is very interesting. During muscle immobilization, mTOR is responded by upregulation in order to compensate for the protein loss. But as we will see in the future slide, that is not effective enough. But this initial transient increase reflects that. But in the end, the expression of FOXO is so strong that overrides the phosphorylation, so it still shows a decrease in the FOXO dephosphorylation pattern that causes part of the muscle atrophy. The two specific proteases, estrogen-1, they are not actually proteases, but they are ubiquitin ligases. It's called E3 ligases. They facilitate the binding of the protein to ubiquitins. And only when the binding occurs, the protease will work. So these two are actually the rate-limiting enzymes in proteolysis in the muscle. You can see that 2 weeks really activates this enzyme in all major muscles, and remobilization, estrogen-1 stays, and MRF1 is still above the resting level. So this is probably explain the muscle protein loss. We mentioned before the mitochondrial dynamics plays a role also in causing some of the deterioration in the muscle. And we can see that during the 2 weeks of immobilization, that the mitochondrial fusion protein showed a steady decrease, whereas mitochondrial fission protein showed a steady increase. This shows that mitochondria probably gets more fragmented during the immobilization period, and this could be controlled in part by the PGC1 decrease, because fusion 2 is a target gene of PGC1 alpha. So the decrease could be explained by other redox signaling pathways. We also looked at the immobilization and remobilization on autophagy, and the two we measured, one is Becklin and one is LC32. Both are the markers of the formation of the autophagosome, which is the initial step to have the protein engulfed by the autophage protein to form a vesicle that is delivered to lysosomes to be consumed. So these are the important markers of autophagy, and we show that an increase during immobilization. And the remobilization, surprisingly, doesn't decrease it, but actually cause a dramatic increase, and we still don't know why. Two other markers, one is P62 and one is the BNIP3. They showed an increase, and we are not quite clear what the dynamics are, but these are preliminary data showing that immobilization could affect the autophagy. So as a summary, disturbance of redox signaling seems to play an important role in muscle disuse atrophy and caused by a downregulation of PGC1 alpha signaling and increased ROS generation, NF-kappa B activation, and also the FOXO activation may be important for the increased proteolytic gene expression. So during immobilization, we can see that the inflammatory pathways is a dominating phenomenon. That is shown by the FOXO activation and NF-kappa B activation, whereas the PGC1 alpha, which possibly influences this process, is weakened. So the predominant force is the inflammation and proteolysis. And again, this is our model, but is there any way we can increase PGC1 alpha expression so that we can inhibit FOXO and inhibit NF-kappa B so that we can preserve the muscle? To show that how we can use the redox signaling to provide some therapeutic benefit. This is by an overexpression of PGC1 alpha through an in vivo transfection method. Since these immobilized muscle cannot be exercised right away, we still think exercise is the best way to upregulate mitochondrial function. But because these muscles are so weak after immobilization, there's some difficulty. So in this way, we try to overexpress PGC1 by a local injection of PGC1. So the procedure, I'm going to simplify that. First, we insert the PGC1 sequence into the E. coli in the plasmid. Then we tried it out in the C2C12 cells to be sure that PGC1 is overexpressed. And then we transfected the TA muscle to be sure it's transfected. Then we find that PGC1 can bind with its target, NF-alpha-1. This is the binding study. And also we go through a reporter gene to show that PGC1 can increase the cytochrome C tag reciprocase. And finally, we verified that PGC1 injected into the muscle is uniformly expressed in the whole TA muscle rather than just being in the injection site. So the procedures after injection, repeated injection of 3-4 times, we have an electroporator to permeabilize the muscle membrane to allow the PGC1 spread throughout the muscle. So some of the quick effects that it's not being published yet, but we showed clearly that PGC1 is overexpressed both in the cytosol and in the nucleus. And we see that NRF1 per se and NRF2 are not specifically improved by the PGC1 overexpression. However, all the major parameters of mitochondrial oxidative capacity, such as cytochrome C and cytochrome C4, COX-4 subunit, TFAM are increased. So this is the control muscle. This is the immobilized by 2 weeks after 1 week of remobilization. This is the control, which means you inject. This is the two open bars are the injecting of the empty vehicle. So there's no PGC1 injected. Those two are the ones that injected with PGC1. So you can see the effect. And the mitochondrial density has been shown to dramatically increase with injection method. So this is the decline, but after the injection, the mitochondrial density is improved, and the mitochondrial DNA replication is improved, and the mitochondrial ATP production rate is increased by the local injection of PGC1. I mentioned before that there's a very large response by the mTOR pathway. We measure the mTOR pathway, and that is indicated as a compensatory way of the muscle try to restore the muscle protein synthesis. However, we did not see any improvement of muscle size, so the muscle size still decreased, and that's not being prevented by overexpression of PGC1. Nevertheless, we show that the two protease-related enzyme, Atrogene1 and MIRF1, which is increased by immobilization, remobilization protocol, is dramatically declined by the overexpression of PGC1. And we show that PGC1 overexpression decreased mitochondria hydrogen peroxide production, but not NADPH oxidase, neither is xanthine oxidase. So it's specific to the mitochondria. And this shows that the protein oxidation marked by 4-hydroxynonenal is decreased by PGC1 overexpression, indicating oxidative stress is reduced. Here we show that pro-inflammatory cytokine levels are decreased. This is the interleukin 6 is decreased, and this is the NF-kappa B activation is decreased by PGC1 overexpression, and we mentioned that this overexpression could inhibit PGC1, but an increased PGC1 could in turn inhibit NF-kappa B and remove some of the negative effect. So you can see that interleukin 6 is decreased by overexpression. This protocol increases manganese SOD or SOD2 activity, but not the other enzymes, such as catalase, glutathione peroxidase. And also, this protocol does not increase the protein levels of SOD2, but only the activity. What is the reason? Because SIRT3, as I mentioned before, is a deacetylase, and when SOD2 is acetylated, it loses part of its activity. So the increase of SIRT3 expression can decrease the acetylated form of SOD. So you can see that the SOD is acylated form of SOD is decreased. That explains the activity increase in the previous slide. So as a summary, PGC1 overexpression by in vivo transfection can stimulate mitochondrial biogenesis and function and decrease oxidative stress and damage, inhibit NF-kappa B activation and inflammatory cytokine expression, and stimulate SIRT3 signaling and increase SOD2 activity. So we can see that redox signaling sometimes could be a negative pathway, cause pathological effect in the muscle, whereas if we take advantage of the positive effect of some of the signaling pathways and utilize their crosstalk mechanism, we can maximize the effect of exercise by overexpression. This is the final slide that I would like to acknowledge many of the students and the collaborators. Also, I appreciate your patience in going through this tremendous amount of information and sometimes confusion. Thank you very much.